Hi, so my project is on phylogenetic comparison between baculoviruses and invert species in different geographical regions. The purpose of this study was to just sort of compare phylogeny to infectious agents. So I began with two specific research questions, one being, how are insects and shrimp structured phylogenetically in different geographical regions? And the second one being, what is the phylogenetic relationship between the GP64 gene in baculoviruses that causes the nuclear polyhedrosis virus, or NPV, in invert species and that which causes infections in larvae and shrimp? So I chose these two species because they are the only ones that are susceptible to NPV as far as my research has shown. They have vastly different environments. One lives in this oceanic biome, surrounded by other crustaceans, other aquatic species, while the land animals such as butterflies and moths, they travel everywhere. So quickly looking at my graphical abstract, I began this with two images, one of larva and then the other of pink shrimp, both affected differently by NPV, control agent later on, which brings up questions of conservation as well. So the purpose of this project was obviously to highlight phylogeny to infectious agents, expressed alongside a host protein at the same time, which is what generates the baculoviruses, especially in invert species. So these two species, their biomes are vastly different. So looking at their previous population, we see pink shrimp are pretty stabilized in population. They're often found in areas of Mexico, like South Mexico, the Gulf of Mexico, then Florida Keys, whereas going back to my previous statement about these land animals, they tend to travel, so the virus will travel with them as well. And so also I just wanted to compare the GP64 gene to the location of the species as well, and just to see if that has anything to do with the effects of the virus. So baculoviruses, they're a group of DNA viruses that attack and infect the species within their cells. So once it's become infected and dies, it's cycled back into the environment, and typically polyhedron is another gene that is considered to be the foundation for these virons. So that's something to consider as well. Use GenBank to collect the species, the accession numbers, and then I used 16S rRNA in the search field just to see if there was any diversity among the species as well. And then I aligned it using MegaX. Pink shrimp are on the more rarer side, whereas butterflies are suspected to stem from moths, and progressively going up. There is a relationship between the two, but it's not, which makes sense. The closest land animal to a shrimp is a pill bug, which is minuscule. And then looking at figure 2b, we see that the baculoviruses all stem from the GP64 gene. So looking at the bottom, we see PMSNPV as the smallest or most independent virus, which kind of makes sense because this is the only one that was found in pink shrimp as well. While the other ones show a much higher confidence the more you progress up the phylogenetic tree. And so there were a few shortcomings just as far as not all strains of the gene were able to fuse naturally with an assisting protein. and there were variations of the severity of the gene. And also, the research does advance the field because it shows a genetic and internal foundation regarding the effects of this virus. So then it kind of brings up that whole biologically controlled agent and how significant that is as well. So when we think about how that's used for pest control in agricultural and forestry manners, it kind of brings up another question as to what's more important as far as wildlife or plants, vegetation. And then that's further that comparative conservation issue as well. And then there are my citations. Thank you.